Good morning, ladies and gentlemen. Welcome to BSW 2023. I am Pooja Sawant. I'll begin with the first session for today. It is titled as Cell-Based Potency Assay Designs and Considerations. The moderator for this session is Ms. Subhrata Khedkar. Ms. Subhrata Khedkar is currently working as an Associate Director at Bioassay. She has completed her MSc in Biochemistry from Devi Ahilya Vishwavidyalaya. Her core expertise lies in bioassays and cloning and expression studies. Ms. Khedkar is well versed with USP and other international bioassay guidelines. She is an approved USP instructor for the bioassay design, development, and validation, and residual DNA testing USP education courses. She holds an overall 15 years of experience in the industry. I would like to welcome Ms. Subhrata Khedkar, and I would request her to begin with the session. Thank you.
Before we begin, let me talk a little bit about bioassays. Bioassays are critical quality attributes for biotherapeutics and cell gene therapy products. For every product, bioassays are extremely important. Now, bioassay development requires considerable efforts, time, and cost. So it becomes extremely essential that when we are developing a bioassay, we should really make sure that they are robust when they are developed. And another question is, what kind of bioassay do we want to develop and when? This is also a very important question. Do we just want one bioassay, or there are multiple assays that will be required for our product characterization? So our speakers will address different aspects of these. So now I would like to invite our first speaker, Dr. Abhishek Mathur. Dr. Abhishek Mathur is Chief Scientific Officer at Biologics Private Limited, and he's done PhD in Biological Sciences from Northwestern University and MBA from Duke University. He has extensive experience in global business, technology and innovation, leadership, CMC development and strategy, product lifecycle management, FDA EMA regulation, operational excellence, data analysis, and AI and ML for Biopharma. He has more than 15 years of experience in the industry. He has 7 publications in his name and a patent on multiplex bead-based assays. In addition to this, I would like to say that of all the bioassay people I have ever interacted, I find he is a very enthusiastic bioassay person, and we are very fortunate to have him here and open this session. So, Dr. Abhishek, over to you.
So, very good morning, and thanks, Subhrata, for calling out the excitement really well. Also, I think ChatGPT has done really wonders in putting my profile together, so thanks to this. But yeah, so yes, bioassays I'm pretty enthusiastic about, but I'm a chemical engineer, and who got into my first role at Amgen in potency assays. I didn't know how to pipe it. I didn't know how cells look like under a microscope. The best I knew was a bread mold, which I looked under a microscope in school in 11th or 12th grade. So I didn't know what I was getting into, and I was very excited. It was my first job. It was a pretty good company, and my manager, Venkat Mukku, who built Bioassays or Potency Assay Group at Genentech and Amgen, he was there to support me. That's what he told me. He told me, in your interview, don't bring cell biology if you have not worked it out. I said, forget cell biology. I have not even had a cell bio 101 class, so I will not bring it up. So that's how I started in there. I think it worked out well, because being the only person who can understand a little bit of math in a group of 25 amazing cell biologists and molecular biologists, it was a great learning from them. I learned my cell culture from Wikipedia, and then I realized after 3 months that all mammalian cells have a very similar cell culture protocol. So, and that helped me, jokes apart, it really helped me in establishing some patterns and going by empirical understanding of this, how bioassays work, how potency assays work, because it's a very skill-dependent exercise. Every pathway that you'll try to mimic in your in vitro system, it will be different for every drug candidate. Later on I realized that it's the only method, analytical method, that tells you that molecule is functional or not. If it is not, then it's a big furor in the whole company that this molecule is not functional, or if you see any differences. So bioassays, it's a relatively smaller community. However, it's a very niche and very unique community, because if you don't demonstrate that a molecule is functional, you don't have a molecule, or you don't have a candidate. So it's important that you understand, you appreciate this uniqueness of the function. You'll always be clubbed with analytical, who determine the purity of your product, but functionality becomes very important. And Shubhrata and we were talking, and she suggested I talk on Fc functionality for IgG-based therapeutics, and I'll call IgGs just for sake of simplicity. And this Fc functionality is a totally different ball game. You talk about the CDR region of an antibody, which is the business end of the molecule. Then FC, you think if you put Fc just to have a greater circulation and a greater half-life, but Fc does a lot more. In 91, when Fc gamma receptors were put on the map, it was published, a lot of literature started to trickle in. You started to realize that there are a lot of ancillary functions that this Fc region is implicated in, effector functions, and this can really impact the mode of your action. So I'll talk about a little bit more, and I have 20 minutes, right? So we'll go with the 2020 T20 style, a little bit faster. This is the most important slide. If you don't take anything away from this presentation, this is the most important slide, and this is extremely important in the industry we are in. We touch real human lives, patients and their families, and it's important that whatever we do, whether it's we are in bioassays or analytical or process development, we need to think that, are we making the right product? Are we doing the right things? Very basic value, are we doing the right things? Because real lives are at stake. So this is important in how we as bioassay people, or as industry biotech leaders altogether, we bring this culture, bring this thought process to every single person who gets into this industry, right from college, right from the fresh graduates. It's important that we talk about this on the day one, because patients first, humans first is important. It's a noble profession. It needs to be. I can't overemphasize this. So moving on to T20 power play time. So there are a lot of biologics modalities that are being developed. It started with MABs, it started with proteins like insulins, then moved into IgG, which was a big revolution, the MABs. And then there were a lot of MAB derivatives, and we have a lot more newer modalities. We talk about BiTEs, we talk about bikes, we talk about oncolytic cell therapy, we talk about cell and gene therapy. So all these things are pretty exciting, and it's very exciting time for us to be in the biologics world, because suddenly all these newer pathways are opened up as the potential therapeutics pathways, and we need to characterize these pathways very closely. And while the commonality of this characterization stays the same, is your product, the physical and structural attributes matching to what you want to have? Is a sequence of a protein-based therapeutic the right sequence? Is the purity right? Is it binding to the right target as it's intended to, whether it's a CAR-T or whether it's a protein? And importantly, again, coming to the epicenter of today's discussion, but epicenter of the biologics itself, does it have the intended biological activity? So one is potency, one is other ancillary activities which may be implicated in the pathways as well. So biology is complex. So your molecule not just does what your Fab region, IgG Fab region is supposed to be doing, also there are tons of other activities it may be involved in. So you need to characterize and understand every functionality to ensure that your product, and there are no surprises in the clinic. So bioassays, as I said earlier, bioassays are the only methods that demonstrate that molecule is functional as intended. In the industry, it is used in the discovery side. I mean, now I'll be talking mostly around protein-based therapeutics and primarily IgGs, because that's the focus with the Fc in there. So in industry, these methods are used in screening to get the right molecule, right candidates, advance through the pipeline and get to the first in human stage. And during product development through commercial, these bioassays are very, very crucial, very crucial to ensure there's a batch-to-batch consistency for this therapeutic, for the product. And it's also used for ensuring that the product is stable during its storage. If FDA doesn't, or regulatory agencies don't have any question on any other attribute, you've covered it well, it's a default to ask a question on FDA, on these bioassays, potency assays. I've always seen, I've worked on tons of molecules. I've always thought that, if everything else is perfectly captured, there will be a question on bioassays. Not because they're imperfect, but because they want to make sure that we understand our product. So it was a great learning because, as an engineer, I was taught to think on the biological aspects and understand at the molecular level, and it was very, very fascinating how these bioassays helps us to learn about this product in general. So very quickly, this is antibody, as most of you know. There's a Fab region, which is usually the business end, the target binding region, and there's an Fc region. Now, in Fc, the upper part of ch2 hinge, that part is implicated in binding to Fc gamma receptors. The lower part is primarily, it binds to between ch2 and ch3. This region is implicated in binding to FcRn, neonatal Fc receptor, which is implicated in half-life modulation. We'll talk about it a little bit. And also to your high mannose. High mannose also impacts, presence of high mannose in this glycan region also impacts your half-life, but again, this is a binding region for mannose binding lectins and other mannose receptors. As I said earlier, each possible biological activity needs to be well characterized, as you make a therapeutic out of it. And you need to understand all these biological activities and characterize it. Some of them exist, some of them don't exist, or they are not active. But you have to demonstrate that, all these activities, you have characterized it and ensure that whatever active pathways are, you are characterizing them and ensuring that batch to batch there is a consistency in that. For biosimilarity, you need to ensure that all these pathways are matching to the innovator product. So Fc functionalities, these are categorized in two different parts. One is the Fc effector functions. So this involves cell killing through different pathways. So this is important because it can impact drugs efficacy and drug safety, because cell killing is important. You don't want to kill your non-target cells, because that's directly impacts patient safety. You want to ensure that if there's an effective pathway which is a part of mechanism of action, it is well characterized and it is consistent from batch to batch, or to your innovator product if you're creating a biosimilar. So this is important, and we'll talk about this a little bit later. There's an FcRn-mediated IgG recycling. So IgG-Fc binds to FcRn. FcRn helps in recycling the antibody within the cell. It brings, it recycles it, and that's how it increases the half-life. So this is a very peculiar pathway, because half-life of this IgG-based drug is increased just by recycling. Is there any other way? Probably. FcRn also binds to IgG-Fc and prevents the glomerular filtration. That's another pathway through which it prolongs the half-life. It is thrown back into circulation. But if you think about it, FcRn binding does not impact safety or efficacy. It only impacts, the half-life, and hence impacts how frequently the drug needs to be administered. So regulatory agencies are perfectly fine in having just an FcRn binding assay to capture that the molecule is indeed functional and it binds to Fc. Your Fc is intact. They are not worrying about having a functional assay that shows the recycling, because it is not related to safety or efficacy. Effector function impacts safety and efficacy very directly. So they are very, very particular about it. We'll talk next. FcRn, very quickly, we talk about the pathway, the recycling pathway. And FcRn binding is pretty standard. It started with the SPR assays long time back. Some challenges happened because of the variability you saw. Now people have moved on to the alpha screen assay or cell-based assay. It was my first project, industrial project ever. And it stuck with me forever, for the right reasons. I did a lot of work on this, how to improve FcRn binding to improve the half-life. There was a lot of work going on that side. But I still feel passionate. That diagram I made in my second month in 2006. And it's a simple binding assay, cell-based assay. People say that cell-based assay is more relevant than a SPR or a solution-based binding assay, but regulatory agencies are accepting both as it doesn't have huge impact on the FCN binding cycle. It's not a safety efficacy assay, so they are okay with that. Now moving on to the effector functions, which are cell-killing pathways impacting safety and efficacy of the drug. We have time, right? Yeah. So these are mediated through 3 different Fc gamma receptors, 1, 2, and 3. And 2 and 3 have different polymorphisms, different variants in the system as well. And what they do is, different variants means different levels of affinities. And what it means is, different affinity means different level of cell killing through these different effector function pathways. It is observed over years that Fc gamma 3A receptor binding is a strong predictor for ADCC, and Fc gamma 2A is a strong predictor for ADCP, which is antibody-dependent phagocytosis, cell-mediated phagocytosis. So this ADCC, I am assuming that most of you know, but ADCC is the most well-characterized pathways, and it is one of the key pathways, for certain drugs. And it is one of the very important secondary pathway for some other drugs. So FDA is very particular about ADCC because it can be a real safety concern if it is not in the primary or secondary pathway for a drug. And phagocytosis, ADCP, it is something relatively newer in the horizon. In the last 5 years only it has become prominent, but it is again causing a safety concern for some of the drugs. And FDA or regulatory agencies want us to characterize these pathways more and more. But yeah, Fc gamma receptor 3A and 2A, in the initial stages of development, agencies are okay in having just a binding assay to demonstrate that there is a binding, but when you get to the BLA or the marketing authorization stage, you want to have a cell-based functional assay to demonstrate that indeed the cell killing through ADCC or ADCP is indeed happening, and the molecule is functional the way it's intended to. The other important component of effector function assays is the presence of glycans. Now, glycans are very difficult product attributes to control. You can, change the cell media. You can have the right strain. You need to have the right process conditions, and still, you may not have full control on glycans. However, people have tried different strategies, putting a small molecule inhibitor as one of your cell media components, to ensure that these fucose levels or galactose levels don't change much and they are in control, because some of these glycoforms, fucose, galactose, and even sialic acid, these are implicated in effector functions. And it's important to control these 2-3 specific glycoforms, including high mannose as well, which impacts, in fact, PK. But these need to be modulated, these need to be controlled. ADCC is very well impacted by fucosylation levels. It's very well known now. It has caused some of the safety concerns for some of the already approved products, and it's an important attribute that need to be controlled way early in your program development. Same thing applies to the alpha galactose and sialic acid as well. This slide just gives you an example that how fucosylation levels, high fucosylation levels can lower ADCC activity. So for biosimilars, this is very important because you need to control fucose levels so that ADCC activity with the innovator product is in fact almost matching. And while you are controlling 20 other critical quality attributes in your process, fucose is one of the toughest one to control, and people have tried different technologies, different process steps to control these levels. Okay? Okay. Yeah. Okay. So some of the companies have their own niche ways or technologies, platforms that they used to control this glycan distribution across to modulate the effector function. And Xencor, Genentech, there's a much longer list than that, but these are the key ones that have been successfully able to modulate that. Now, moving on to a specific ADCC assay, which is the most important, or I would say most well-characterized effector function. And the traditional format is a primary cell assay. I think a lot of may have been aware of this ADCC assay, but it's highly variable because it's a primary cell assay. There's a donor-to-donor variability. Traditional layouts, readouts can, it's a cell-killing assay, so dye release or cell proliferation, these can be the readouts. But higher donor-to-donor variability, keeping cells in culture for more than 2 days, all these things are very challenging. So people have started to use engineered NK cells, for cell-killing assay. But engineered NK cells, again, do they have the right level of expression or they are overexpressing? All those things, considerations become very important. People are using binding assays, bridging assays, and these are very variety of different formats that have come across as a surrogate assays. But the important aspect is you need to ensure that the mechanism of action stays intact and it is clinically relevant. It is a right assay because overexpression might lead to higher sensitivity, but in vivo it may not happen. So this is crucial. The clinically relevance and representative MOA with the right cell lines is the most important attribute for these assays. This slide does talk about a panel of assays that have been successfully used for any given product and which actually works out beautifully in your BLAs, in your dossiers. Things are moving, regulatory agents are moving towards, pushing you towards more functional assays, especially for the BLA stage. But some of the assays in the initial developing, the binding assays and all those things, there are around 12, 13 different assays that you may have to run to characterize this Fc functionality very well. So a quick case study. In 2012, Celltrion got approval for Remsima as a biosimilar to Remicade. Everything, all the biosimilar attributes were matching: analytical comparability, potency activity, clinical comparability in rheumatoid arthritis patients, but there were some differences observed in the fucosylation levels, and hence some differences in the in vitro ADCC assay that they had. These differences led to different regulatory positions by different regulatory agencies, and FDA was not happy to see these differences in ADCC assay. So the approval got delayed. They were not ready to approve it. It was a big furor with Celltrion and across industry. And Amgen was working into biosimilars, and I was at Amgen, and we were working on NK92 cell-based assay, killing assay, and we realized the importance of that way before that, and understood that understood FDA's position to that. And we are pretty satisfied that the way we were approaching Fc functionality stood intact. So what Celltrion did was, based on the feedback received, they modified their in vitro ADCC assay and used the physiologically relevant target cell lines and effector cell lines. So as I said earlier, if you have overexpressing cell lines, it can be challenging because you have higher sensitivity than required. You might see an ADCC activity differences that may not exist in vivo because physiologically those cell lines may not be too relevant. So they modified the ADCC assays. They ran those assays with the relevant target cells and whole blood just to prove that, or just to understand rather that there are no differences if the molecule goes in vivo. And no data indeed showed that there was no ADCC activity with relevant target cells. And they observed similar activities with the primary whole blood cell assay. Based on those results, finally, after some delay of around a year or so, FDA approved the drug, and it became the first biosimilar drug approved by EMA and FDA. It was a great success story how Celltrion overcame that. I have no stakes in Celltrion, by the way, so this is a great case study for all of us in the bioassay world. So next 3 slides, I quickly brush through this FDA's comments that I've seen they're making around this Fc functionality assays. In one of the cases, these are real FDA comments, by the way. So in one of the cases, the sponsor made some Fc mutations, but did not showcase that how it improves your Fc receptor binding or functionality. So they asked to provide us the data demonstrating that mAb works as designed, and the changes are in fact working out the way they were intended. Second example is around the primary MOA mechanism of action was ADCC, but the assay was in the ELISA format of just FC or CDR binding ELISA. But FDA came back and say, hey, you need to have at least a bridging assay that shows that both sides of your antibody are binding to target and your Fc gamma receptor. And for BLA, they definitely want to see a cell-based functional assay, a cell-killing assay. The last example where the sponsor observed the reduced affinity for Fc gamma 2A, which is a stimulatory receptor, but they did not show any binding to other Fc gamma receptors, especially Fc gamma R2B, which is an inhibitory receptor. So they asked the sponsor to have a second release potency assay for Fc gamma 2B receptor as well. So I mean, it demonstrated that FDA or regulatory agencies are pretty serious in having these different Fc gamma receptors, the binding, and show it functionally that the binding is not causing any adverse signaling in cells, or if it is indeed implicated in the pathway, they actually are implicated in the right pathway. And characterizing just one receptor versus other may not work. This example perfectly shows that. So you have to have this panel of around 12-13 assays, demonstrate very early in the program how this binding helps, or if the pathway indeed exists or functional, and then as you can use it for screening purposes. You can get into once you get to the later stage, these learnings will be very important as you design or engineer the antibody as a therapeutic. So last slide, a summary slide, various Fc functionalities exist, and they need to be well characterized and understood. Just characterizing the Fab region or CDR region may not be enough, is not enough, not just may not be, is not enough. We need to understand our product, that's the bottom line. We need to screen for ADCC or effector function activity very early in the program and used as a part of our screening checklist. Always be cognizant of your assay system relevance. Your assay should not be more or less sensitive than how it would be in vivo. So you need to choose the right cell lines, you need to choose the right assay format, and I can go, I have a totally separate talk I can design on this, because FCR and also we have seen some assay artifacts, a lot more sensitivity to 0.1 level of aggregates, which was not the right format. And always strive to have a cell-based functional assays. I think I cannot overemphasize this enough very early in the program. It's difficult, it's expensive, but start working on this very early in the program so that you have fewer surprises later in the game. I think that's it from my side. I'm on time, right? Slightly on. Okay.
Thank you so much, Abhishek. We'll take one question, and I saw your hand first.
Hi Abhishek. Hi Abhishek, good morning. So, myself Ravish Patel from Sun Pharma, and your talk was really wonderful, nicely compiled, and especially the last part of the FDA things is really encouraging. And I have a quick question. So, have you came across any time that FcRn binding improve or decrease based on the glycan modification? Like, does any glycan play any role in FcRn binding? You have any publication or any paper you came across, or you have some experience prior?
Yeah, so conventional wisdom is glycan, because glycan is located in the hinge region.
Yeah.
It may not impact FcRn binding, and we have seen the same way across a variety of molecules. Having said that, there are some hinge mutations that you can make that improves FcRn binding, but it's not related to the glycans per se, in my experience. And I think there are a lot of literature published on that.
Because like fucose and other glycan play the role in functionality. So because see, in EPO, sialic acid play a very important role in clearance, T half-life, correct? So I have never seen such things in like a circulatory T half-life and the correlation of the glycan, especially in a MABs. So that is only the things which I wanted to know. And the second question is that you have also mentioned the bisecting glucnec. So bisecting glucnec, I think if I am not wrong, bisecting glucnec is not there in a CHO cell line. It is not present in CHO cell line, if I am not wrong.
So again, there was a very generic presentation of the analytical, but or the glycan, glycophore.
No, just I just wanted to understand, because what I have having the knowledge that it is only present in a SPO2 cell line. So it doesn't have, but if you have something related to this, I would be very happy if you can share after this.
We can discuss it offline, but
some paper or something which, because I wanted to understand the bisecting glucnec role in the potency assays, like how it plays an ADCC and CDC, because what my understanding is it is not there. So I will be very thankful if you can share something on that.
Sure, we can
Yeah.
surely discuss
Okay.
offline. But yeah, the point is the glycoforms can have impact on this FC effector function.
Yes, yes, glucose.
Not
and
on
fu-
FcRn.
Yes, yes, yeah, correct.
Not on FcRn per se, but again, it's an evolving game, and high manos impacting PK is the new regime that is being
Yes,
developed right
yes.
now, understood. So 5 years from now, I'm expecting this, high manos binding assays would be a norm, especially with map-based therapeutics.
Okay, thank you very much.
Thanks.